Okay, so thank you, Ina, for the introduction, and thank you to the organizers for the chance to speak today. It's a very exciting and fun opportunity to present my work in the lab at MIT. So in my postdoc, I ask a fundamental question in molecular evolution, which is about cellular novelty and how do new genes emerge when organisms adapt to different environments and challenges in evolution. And we know that in nature, there are a few mechanisms by which genes can emerge, if it's gene duplication and divergence, or horizontal gene transfer, or protein domain shuffling. However, I am interested in an additional mechanism that I find very intriguing, that is termed de novo gene birth, in which new genes emerge from previously uncoding loci in genomes. And the idea here is that these uncoding loci are somewhat expressed, generating random small proteins that are inside cells. And if those random small proteins are beneficial and perform some function, a natural selection can then optimize them until they become fully functional genes and join the rest of the genome. And we believe that there are some key questions that are unresolved and are interesting to ask in this mechanism, about this mechanism. So the questions we are asking are, what is the likelihood of such small random proteins to actually be functional? If they are functional, what kind of different challenges can they help cells solve as they evolve? And what are the molecular mechanisms that these random proteins can assume in cellular systems? So to study these questions, I have come up with the following experimental system in which I've constructed a big plasmid library in which each plasmid is harboring a unique gene with 150 random nucleotides, basically meaning that each plasmid is expressing a unique random protein. I transform this plasmid library into E. coli and then challenge this library with two different evolutionary challenges. One is the toxin MASF, which is an endoribonuclease that degrades the transcriptome, and the other is the T4 phage. The reason we chose these two challenges is first that they are very different. One is a specific protein function, and the other is a complex biological system. So we thought we would get different functions from these random proteins if we use these two challenges. The other reason is that they just allowed us to very easily find functional proteins, because if one of the proteins is able to inhibit MASF or T4, then those colonies will just be able to go on plates and we will be able to detect them very easily. So we now show you the results of the MASF screen and then move on to the phage screen. We found two random proteins that were able to inhibit MASF. Here you see one of them, which is termed RAMF for random antitoxin of MASF. And here you can see a serial dilution of a bacterial culture. So lots of cells are seeded here and less cells are seeded here. And when MASF is expressed as expected, cells cannot grow, their growth is inhibited, and the plate is mostly empty. However, when RAMF is expressed alongside MASF, then the phenotype is rescued and the cells are able to grow. Interestingly, this is a MASF-specific defense, because when we tried RAMF against a different MASF homologue, ChIP-B, it was unable to inhibit it. We then wanted to understand more about the mechanism behind this MASF inhibition by RAMF. And the first thing that we tried was to follow the stability of the MASF protein. So if we stop translation and follow with time, follow MASF expression level with time, you see that in an empty vector control, MASF is very stable as expected for this type of toxin. However, when RAMF is expressed, then you see a rapid MASF degradation, which could be a cool mechanism for how MASF is inhibited if it is simply being now degraded. It was quite hard, but we also were able to find the protease that is responsible for this rapid degradation. So it turned out to be FTSH. And you can see here that upon deletion, this deletion of this protease, MASF levels went back up, even if RAMF was expressed in cells. Next, we wanted to understand more about how does RAMF promote rapid degradation of MASF. So to do so, we performed an IPMS experiment in which we pulled down on RAMF and send the sample for mass spectrometry to understand what other proteins RAMF interacts with in E. coli. And among the top hits that we got, we saw the cellular chaperones GROEL and DNAK, central chaperones in bacterial cells. This led us to the following model, in which unfolded MASF interacts and rely on cellular chaperones to fold correctly into its native form that is toxic and hence results in growth inhibition. But maybe when RAMF is expressed, these chaperones are occupied and that this route is blocked for the unfolded MASF. So instead they are being sent for degradation by FTSH, leading to cellular growth. One prediction of this model is that if we overproduce these chaperones, that even in the presence of RAMF in cells, the unfolded MASF is still able to find some free chaperones to fold correctly, and its toxicity should be restored. So this is exactly what we see. And you can see here that when we express MASF, RAMF, and overproduction of these chaperones, toxicity of MASF is restored. Another prediction is that even if RAMF is expressed, but we are somehow able to interfere with its interactions between the chaperones and itself, then that should also restore toxicity of MASF. So we used the AlphaFold to model the interaction between RAMF and DNAK. And when we modified and mutated the positions that AlphaFold predicts that are important for this interaction, we got a defective RAMF version that is able to inhibit MASF at less efficiently. So we've used this data and other data that I don't have time to show you. We concluded that the random protein RAMF is able to somehow modify the cellular physiology. And we also showed some of how it can do that by interacting with cellular chaperones and affecting protein degradation. All of this to promote cell survival and adaptation under the evolutionary challenge that we presented to cells. I will now move to the second screen against the T4 phage. And this screen was done with a friend of mine from the lab, another postdoc, Chris Vassallo. And here we actually found dozens of random inhibitors of phage, which we termed RIP. What you can see here are plates in which we co-incubate phage with bacteria. So in an empty plate, like in an empty vector control like this, no colony is able to grow on the plate and we get an empty plate because the phage is simply killing all of the cells that we co-incubated with it. However, when RIP-A, one of our random inhibitors of phage, is expressed, you can see that colonies are able to grow because presumably RIP-A is helping them, is providing a T4 defense. Interestingly, we found that this is not a T4 specific phenotype because when we tried other phage against this protein, for example, here you can see lambda-vil, it was also able to defend against them. I don't have time to show you, but what we found is that RIP-A is leading to the activation of a very known, well-studied signal transduction pathway in bacteria termed RCS. And activation of this pathway is leading to the secretion of capsular polysaccharides that are coating the cell membrane and blocking the phage from absorbing well to the phage and basically inhibiting the initiation of a viral infection cycle. And indeed, when we delete the RCS pathway, RIP-A is losing its ability to defend against T4 phage. In addition to that, we also found another class of RIP proteins that are able to provide a T4 specific defense. So you can see here that colonies are able to grow against T4, but are unable to do so against lambda-vil. And as expected, they show an RCS independent phenotype. We always ask what is the mechanism in the lab, that is our thing. So what we found is that RIP-B is leading to a reduction in OMP-C levels. It's a very significant reduction in OMP-C level. Why is this relevant? Because OMP-C is actually the receptor that mediates T4 adhesion to bacterial cells. So presumably when you have less of this receptor in the membrane, it is harder for T4 to find cells to infect. With that, I would like to end. I hope I convinced you that random proteins can have meaningful, very interesting functions in bacterial cells. I showed you that I screened my library against two very different challenges, and I still found functional proteins that were able to assist cells to survive these challenges. I showed you that the cellular functions that we have found relies on integration and regulation of existing cellular pathways in E. coli. So whether it is interacting with chaperones, affecting protein degradation, or activation of single transaction pathways, these random proteins were able to do so in a beneficial way. And I also showed you that they, excuse me?
1 more minute.
Yeah, I'm finishing. I showed you that these proteins are able to regulate expression levels of specific proteins in E. coli. We believe that this might provide a clue for the evolutionary origin of small proteins that are attracting a lot of attention in the literature these days, and we see many of them with different functions, and it is interesting to assume that, or to guess whether or not they have evolved from de novo in a de novo way. And I leave you with the fact that maybe this is just the beginning, because I have screened only a tiny portion of the landscape of random proteins impossible, which means that we believe that if we screen more proteins under more challenges, we might find more and more functional proteins. And this has vast implications both in basic science and maybe in the industry. And I'd be happy to speak about this and other future plans that I have offline. With that, I will really end by thanking Mike for being a great mentor, Chris for being a wonderful collaborator on the phage story, and to everyone of you for listening. Thank you.